I will be talking about potential applications of mesenchymal stem cells and its secretome in regenerative medicine. So, as we all know, this mesenchymal stem cells, they are multipotent progenitor cells, and they can be isolated from various sources, and they can differentiate themselves into various cells like the osteoblasts, which make our bone cells, chondrocytes, which make our cartilage cells, and adipocytes that make our fat cells. So these mesenchymal stem cells are being extensively studied everywhere in the preclinical settings and clinical settings as well, due to their ease of isolation and the less immunogenic response that they hold. So their multipotency also is a very key factor that helps the clinicians or the basic scientists to apply these agents in a range of cellular therapies, including tissue engineering and regenerative medicine. Basically, these progenitor cells, mesenchymal progenitor cells, they uh, they were isolated traditionally from the bone marrow as of now, as of recent reports, but due to the scarcity of bone marrow, as well as the invasive procedure, the ethical concerns involved in the procedure, scientists have been trying for alternative sources of mesenchymal stem cells from a variety of tissues, which range from umbilical cord, cord blood, peripheral blood, liver, fetal liver, lung, adipose tissue, skeletal muscle, then other amniotic fluids, circulatory system, and synovium as well. So these mesenchymal stem cells, why it's so important? Because it's very, very rare. They occur in 1 in 1 lakh frequency in the bone marrow. And they should be actually culture expanded to a sufficient number so that their research and therapeutic applications both can be validated. Why my lab actually was involved or interested in this stem cell research, or rather the umbilical cord stem cells that I will be briefing today, is because this umbilical cord is a very, very accessible source. It has very less immunologic responses, ethical concerns, and there is no invasive, extra invasive procedure. So these MSCs that are present in the umbilical cord, its tissues, Wharton's jelly, as well as the endothelial vein present, is very, very proliferative in nature. They have a fibroblastic like morphology. They display the mesenchymal markers, and they can also differentiate into the fundamental lineages. But these investigators, they report various sources till date: bone marrow, umbilical cord, cord blood, adipose tissue, menstrual blood, synovium, and many other extraembryonic membranes as a source of these MSCs, which all researchers have either patented or they have followed various different isolation strategies and expansion methods, which to characterize these MSCs from the primary tissue, which is increasingly difficult to compare and contrast the study outcomes, because they display different characteristics, as well as this difference in the characteristics of MSCs from various sources. They hindered the progress of a unified outcome in the field of research. In this regard, those who are working in stem cells, especially mesenchymal stem cells, they always will be referring to this paper, which is a position paper in ISCT, International Society for Cellular Therapy Journal, where they have defined the minimal criteria for defining the multipotent stromal cells, in the sense so that all the researchers who are using various sources of the stem cells can come to one conclusion and identify or characterize their stem cell population from a primary culture, which is very, very difficult basically, and which differs in which the outcome differs even if they have used various sources, but ultimately the cells are mesenchymal stem cells, but the outcome varies based on the source that they have isolated and how they have characterized and how pure that population is. So these two papers are very much important. One is from my old research in Karolinska Institute, the second one, and the first is the position paper, where they have given the minimal criteria that should be met by the researchers so that the cell can be told as a mesenchymal stem cells. Now I come to the secretome and its therapeutic application. What we will be denoting is not only the stem cells, the secretome or the conditioned medium in which the cell is cultured, and from that we tell it as MSC secretome, or in which there are a huge possibilities of presence of growth factors, as well as the extracellular vesicles or exosomes that my lab is interested upon to characterize these exosomes and their benefits in regenerative medicine. So as you can see in the model, this is the MSC, and it has the MSC markers like CD90, CD73, and CD105 as positive, and CD45, 34, and 14, which are basically hematopoietic markers or blood lineage markers, which are generally negative in MSCs, maybe it from any source, bone marrow, cord blood, adipose tissue, and other extraembryonic membranes. And this MSC itself, as well as its soluble factors, which we tell it as secretome, which they release in the culture medium, which we term as conditioned medium, has a lot of growth factors, and they have the capability of immune regulation. So my lab is interested both on the applications of this primary MSCs from umbilical cord, as well as the conditioned medium, which has the secretome, to see its specific therapeutic outcomes in various field of regenerative biology and as well as colon cancer. So basically there are a lot of papers that have explained the superiority of this conditioned medium and the mesenchymal stem cells, various options like bone repair, neural repair, and many others. But a very standardized protocol to characterize, to obtain first this MSCs from a tissue, and then to characterize this extracellular vesicle or the exosomes in the secretome is very difficult, and lot of studies need to be conducted and are still very scarce. In this point only, I got the grant from DST-SERB to check about or to validate the methods to isolate stem cells from umbilical cord blood, to characterize its secretome and to see its effect in the home body, as well as the tumor niche in a colon cancer model that I will be explaining in the recent slides. We have also published a concise review on whatever I told on the secretome of EMSC derived mesenchymal stem cells, which has already been shown to have or exhibit anti-photoaging effect. It can accelerate wound healing. It can suppress ROS generation. It can also cause the cells to differentiate, or in some cases, it can also induce apoptosis. So the preliminary aim of the study was to isolate, expand, vividly characterize this human umbilical cord derived mesenchymal stem cells, the MSC condition medium from the stem cells, which may be very, very crucial if we can characterize, and it can lead to further translation of therapies at various disease conditions. So I will be discussing in very brief about the outcomes that I have started. Some are published and some are ongoing. So we studied the effects of this direct and indirect co-culture of the stem cells and colon cancer to check the paracrine factors which the cells induce on each other once they are co-cultured, exactly resembling the tumor niche of a colon cancer in humans. Another model is on the role of this conditioned medium on proliferation of colon cancer cells, whether, and the role of conditioned medium in Hackett cells, which is a normal skin cell, and also in transdifferentiation potential of the stem cells with signaling pathway inhibitors. In that case, we only my lab is mostly concentrated on pathway, which is very, very important in cell fate determination. So this is basically cord that we get from our Chettinad hospital immediately after the delivery. We prefer the cesarean delivery, and this is the umbilical vein and two arteries which are sterile, like in aseptic conditions, they are excised away. And this is the cord, this is the donor, and from there we excise the two umbilical arteries and one vein aseptically within 6 hours of cesarean delivery. And we take the tissue and mince the sections, as well as we take the Wharton's jelly, and the plates are kept in special stem cell culture medium for 2 weeks. After 2 weeks, we can see this is a explant. This method, what we follow, is already been reported in stem cell research by my lab in Italy, where I was working on this model. And this explant method is really very, very good, where we can get a very pure culture so that other cultures or hematopoietic stem cells are not hindering the characterization of the stem cells. So this is the explant that we get, and we can see after 2 weeks of culture, huge amount of cells can migrate, which has a fibroblastic morphology from this explant. So we take up these cells and we get a pure culture at passage 1, and we take these for various other culture experiments. We firstly check their clonogenic potential of CFUF assay, which is very, very high in MSCs, and they form a very concrete and tight colonies after 15 days of culture. We also check the umbilical cord about their morphology or the cluster differentiation markers, so that we found that these are very highly positive for CD73, CD90, CD105, CD166, integrin beta, and they are negative for c-KIT, CD44, and HLA-DR, which are specifically the hematopoietic stem cell markers. We also found the pluripotency genes OCT4, SOX9, nano were very high, and the stem cell markers were also very high, as well as Wnt, canonical Wnt was also very high in the cells. We also studied some gene expression of some MSC-specific genes like nestin and fibromodulin, which are specifically reported to mark the primary MSCs, and also the proteins, proliferation-related proteins like cyclin D1, P21, and CD6, which was high. We checked the hallmark ability of for where we found the cells differentiated readily within 3 weeks of culture in osteogenic medium, which is 1-cosa staining. Then the alizarin red staining or the oil ore staining for the adipocytes. You can see these are the control cells, and these are the stem cells that are differentiated in the fat cells. What we tell is that adipocytes very strongly, as also differentiate chondrocytes. These are the chondrocytes that make our cartilages, and they are positive for the tolvadin boost, the negative staining of the ACMSCs, which was a really pure culture of MSCs. Then we targeted on preparing and collecting the conditioned medium as a standard protocol patented by our lab, and we characterized this through P1 through GCMS, and we have found 43 different proteins present in this at a very preliminary level. The samples have been given for LCM, SMS, and mass spectra, where results are awaited. We also did the cytokine profiling of the secretome, and we have found this with a QIAGEN kit. We have done the cytokine profiling, where we found that very high level of CXCL1, GM-CSF, GCSF, TGF-beta, CCL4, and IL-8 was very highly expressed in this conditioned medium secretome, and we are characterizing these exosomes with a QIAGEN kit. And we found that when we isolated or purified the exosomes by the standard protocol in our lab, we found that they are within 100 nanometers, which 30 to 100 nanometer in diameter. Excuse me. So these were also very positive, that the exosomes were really high in the frequency. We also checked or screened highly expressed microRNAs from HUMSC as well as the secretome, and we have found abruptly high microRNAs like 21, 125, 146. Mostly they are the tumor suppressor microRNAs that are very highly regulated or expressed in the secretome. And this is the heat map of this. These are done by the QIAGEN microarray cards. And now I will share some experiments on the stem cell fate, how this UCMSC can modulate themselves based on the microenvironment they are in. The first study is on the MSC-derived conditioned medium on HACAT cells. We just treated the HACAT cells with this conditioned medium, and we found that there is a cell strap formation. This has been published in Journal of Clinical Physiology and Pharmacology last year. And we can see these are the normal HACAT cells, and these are the cells when they form the strap formation, which is very much, and after 2 weeks of culture in this conditioned medium, cells are seen to form the HACAT cells, transform them into adipocytes, which is very much surprising. And we can see this will be a very good research in which the HACAT cells, already a terminally differentiated cell, can de-differentiate themselves in the presence of conditioned medium into fat cells, which will be very good to study wound healing capacity, as well as burn patients can be given with this therapy where skin cells can easily differentiate into the HACAT cells. So now we finally targeted one signaling pathway, which is very, very important in this stem cell population. That is the Wnt signaling pathway, which is involved in a multitude of developmental processes, which maintains adult tissue homeostasis, cell proliferation, and differentiation, migration, etc. What we did, we uh, we inhibited our stem cell population with the inhibitor and proliferator of the stem cells, in which we use quercetin, which inhibits the Wnt signaling pathway, and we give lithium chloride, which activates the Wnt signaling pathway. And we found in the graphical abstract is represented that these cells, once the Wnt signaling pathway is inhibited, they can differentiate into osteocytes very readily, and once they are activated, they can proliferate to cells which are exactly like the stem cells in a very, very high rate. So the Wnt signaling pathway was very important. This is another fraction of culture where we treated our colon cancer cells with this condition medium. This is a control cell. As you can see, this cell is after 15 days of treatment in co-culture condition medium, where the cells started shrinking and the colon cancer cells were seen to be dying. However, after 1 month, certain sets of strategies where can be seen that colon cancer cells have started changing their morphology towards a calf-like phenotype. This is unpublished data and ongoing work. We are characterizing what exactly these cells are. It may be the TAMs, tumor-associated macrophages in the microenvironment, they can change, or the CAFs, cancer-associated fibroblasts. Yet it's not clear. It's under the more detailed study is ongoing in my lab now. And we also found that with the co-culture condition medium, the cells or the condition medium, the cancer, colon cancer cells, not only are shrinking, they have less VEGF expression, they have less MMP expression, 2 and 9, which are very pertinent or very high regularly in the colon cancer cells. So this is another model where we are studying, which is funded by DST SERB, where we are seeing the co-culture of resembling the exactly the tumor cell niche of the population of the cells, where the cells, stem cells and the cancer cells are co-cultured together, where we have seen that the cancer cells target the mesenchymal stem cells, and they attach on each other and use as a substratum and grow well. We also are targeting the Wnt signaling inhibitors in this signaling pathway. And I'm just rushing off the slides because of shortage of time. This is another set of experiment where we induce transdifferentiation, very unique property of mesenchymal stem cells, again targeting the Wnt signaling pathway with quercetin and niclosamide, which is in the verge of revision in the journal, where we found that these cells can easily become neuronal stem cells. After 7 days of inhibiting of Wnt, the morphological change was very, very evident, and we found that these cells were diverting towards a neuronal lineage once they are treated or hugely inhibited on the Wnt signaling pathway. This is a summary of the work in a paper that we published, where we have seen that Wnt signaling pathway, when we target these small molecules, they can govern the stem cell fate and diseases very readily. And also the tumor specific niche in the colon cancer progression is a very, very novel therapeutic target, where we can target specific compartments of the colon cancer in emerging therapies of the microenvironment, where both the mesenchymal stem cells are there as a fibroblast and the colon cancer cells are there, and they have paracrine effect on each other, which can be targeted for a future therapy. So this is one of the recent papers that we published in Regenerative Medicine, where we have summarized the sources, isolation strategies, the therapeutic outcomes of the secretome or exosomes at a glance from various sources. And now finally, what we conclude that this UCMSC through the explant method is a very, very novel technique, and we can easily isolate the stem cells. We can culture expand them. They are functionally, phenotypically, and molecularly MSCs. They have a high MSC lineage markers. They show migration potential, wound healing potential, and we can see that the secretome in various disease models, that the soluble factors of the secretome are very, very pivotal in modulating the Hackett cells to become the fat cells, which have immense translational value. Our studies also highlight there is an evident interaction between the stem cells and the colon cancer cells, whether it's a direct culture or an indirect culture, which affects the progression and metastatic potentials. And also, modulating Wnt can aid in change in stem cell fate and might also enhance their transdifferentiation potentials of the cells. So these are the two books that we have recently published with Springer Nature and Elsevier on stem cells and aging, and another on cancer stem cells. I will request everybody to go through the book. It's very good. From all around the world, the authors are there. And these are certain of my publications on stem cells, colon cancer and its secretome and its effects as therapeutic interventions. And finally, we have applied 3 patents also on the applications of the stem cells secretome, its trans differentiation potential. And finally, I will like to